Hello everyone, welcome to the new session of Genetics and Plant Breeding, Cell and its Structure. So this is the fifth part for the cell and its structure. Okay, so in this part we are going to study about the ribosome and centrioles. Okay, so ribosomes are the granular structure. So they are the granular structure and first observed under the electron microscope, though this is the very high power of microscope based upon the bombardment of the electron. Okay, so this is a very important microscope, electron microscope. So that electron microscope enable us to see very small organelle of the cell, that is ribosomes. Okay, so they are the dense particles and first of all seen under the microscope by George Palade. Okay, so they are composed of ribonucleic acid, that is the RNA, and some proteins also, and are not surrounded by any membrane structure. So they are not bound by any membrane. The eukaryotic ribosomes are 80S. Okay, so eukaryotic ribosomes and prokaryotic ribosome differ. Okay, how they differ? Because the eukaryotic ribosome is of 80S type, while the prokaryotic one is of 70S type, right? So each ribosome has two subunits, larger and smaller subunit. We can see that they are made up of two subunit, larger subunit and the smaller subunit. So if the two subunit of 80S ribosome, so here it is 80S ribosome. So this is the 60S and this is the 40S, means the larger subunit is made up of 60S and the smaller one is made up of 40S. So this is the 80S ribosome present in eukaryotes. Right? Okay, so let us see about the prokaryotic ribosome. So this is again made up of the two subunits. Okay, and this is 70S ribosome. This is 70S ribosome. So this is called as prokaryotic ribosome. Okay, now they are also having two subunits, smaller and larger. The larger one is of 50S type, whereas the smaller one is of 30S type, right? So this type of ribosomes are present, that is 70S are present in the prokaryotic cell, as well as they are present in mitochondria, as well as chloroplast. Okay, so they are present in prokaryotic cell as well as they are present in eukaryotic organelles like mitochondria and chloroplast. Okay, so as we see that we are writing 70S type, then 80S type, okay, and their subunit are also having 50S, 30S, 60S and 40S respectively. So why they are having this S type? S means Swedberg's unit. Okay, that stand for the sedimentation coefficient, that is indirectly a measure of the density and the size. So they are denoting the size depending upon the density. Okay, so here we can see the larger and the smaller subunit. So the 70S and 80S ribosomes are composed of two subunits, and these subunits are joined at the time of protein synthesis, and they require magnesium ion. At the time of joining. So this is very important. Next, we are going to study about the cytoskeleton, an elaborate network of the filamentous proteinaceous structures consisting of the microtubules, microfilaments, and the intermediate filaments are present in the cytoplasm is collectively referred to as cytoskeleton. So they are the three different types of filamentous proteinaceous structure that are constitutively referred as cytoskeleton. So there are three different types of filamentous proteinaceous structures. Number one is microtubules, number two microfilaments, and the third one is the intermediate filaments. Okay, the cytoskeleton in a cell are involved in many functions such as mechanical support. So they are responsible for the all the three types of filamentous structures, that is the microtubules, microfilament, and intermediate filament. They are responsible for mechanical support, motility. So when the cell is moving from one place to another, so there is an involvement of these filamentous structures. Okay, maintenance of the shape of the cell, they are also responsible for the shape of the cell. Okay, so these cytoskeleton structures are very important. Cilia and flagella. So cilia and flagella are the hair-like outgrowths of the cell membrane. So on the cell membrane, we can see some of the cells are having cilia, whereas some of the cells are having flagella. So in the case of some of the bacteria, we see that they are having flagella, right? And cilia, in which organelle we see the cilia on all over its surface. So if we see slipper animalcule, that is the paramecium, we see that their surface is having cilia. All over its surface is having small hair-like structures that are called as cilia, right? So they help in locomotion. Right? Cilia are the small structures which work like oars, causing the movement of either the cell or the surrounding fluid. So here in the case of this is the gullet, or you can say the mouth in the case of paramecium. So what happened when this cilia rotate? Okay, all the surrounding water near the gullet having some food molecules makes its way, so that this food molecule come inside this gullet, right? So they are making the surrounding fluid to make a movement in such that the food present in the liquid comes inside its mouth. So in this way, they are very important, these cilia structure. Flagella are comparatively longer and are responsible for the cell movement. Okay. Prokaryotic bacteria also possesses flagella, but these are structurally different from that of the eukaryotic flagella. So there is some difference between the eukaryotic flagella and the prokaryotic flagella. Okay, some bacteria also possesses flagella, but prokaryotic flagella and eukaryotic flagella are quite different. The electron microscopic study of the cilium or the flagellum show that they are covered with the plasma membrane. Very important. These cilium and flagella are also having the plasma membrane structure like this, right? So if this is a flagella, then they are continuous with this cell, right? Like this. And there is a basal body. There is a hook, and this is the filament structure, the main body. Okay, their core, called the exoneme, possesses a number of microtubules running parallel to the long axis. So this there is a covering all over the cilium and flagella that is made up of the plasma membrane only, and their core structure, that is the exoneme, possesses a number of microtubules running parallel to the long axis. So these microtubules are present all over inside the core structure in the form of exoneme. Okay, so exoneme usually has a 9 doublets. They are having a 9 doublets and radially arranged peripheral microtubules. So 9 doublets. If we cut a cross section of a flagellum, then we can see doublet structure like this. 1, 2, 3, 4, 5, 6, 7, 8, and 9. So these are the doublets made up of the microtubules, right? Made up of microtubules. Okay, and a pair of centrally located microtubules. So we are having in the center. Okay, so this is the 9 plus 2 arrangement. 9 plus 2 arrangement. Such an arrangement of the exonemal microtubules is referred to as 9 plus 2 array. Okay, so this is very, very important. Again, the exoneme has usually a 9 doublet. So here we can see if we cut any cross section of a flagella, then we can see that at the periphery they are having a doublet structure. Okay, these doublet structure are made up of microtubules. Okay, and in the center also there is a pair of these microtubules like this, right? Such an arrangement of the exonemal microtubules is referred to as 9 plus 2 arrangement. So here we can see the structure, right? So the central tubules are connected by bridges and it is also enclosed by the central sheath. So here we can see this red color in the figure, central sheath covering the central microtubule, which is connected to one of the tubules of each peripheral doublets by the radial spokes. So here the outer one, that is the doublet structure. This doublet structure is arranged or attached to the central one with the radial spoke. So this is the radial spoke. Okay, this is the central microtubules, and all these 9 doublets are interconnected. They are connected with the interdoublets bridge. Okay, so this is very, very important. This is the internal structure of the cilia and the flagella. Thus, there are 9 radial spokes because there are the 9 doublets. So, all these are arranged centrally. They are attached centrally with the help of 9 radial spokes even. The peripheral doublets are also connected by the linkers. Okay, both the cilium and flagellum emerges from the centriole-like structure which is called as the basal body. Now, the centrosome and the centrioles. So, centrosome is an organelle usually containing 2 cylindrical structures that are called as centrioles. So, if we have seen inner structure of the flagella, in the same way, they are having this centrally two cylindrical structures, okay, and they are present like this. They are not surrounded by any membrane like structure. So this is called as centrioles. Centrioles. They are surrounded by the amorphous pericentriolar material. They are surrounded by the pericentriolar material. Very, very important. Okay. So centrosome and centrioles, these are the organelles usually containing two cylindrical structure called the centrioles. So this is the two cylindrical structures, okay, called centrioles. And this unitedly called as the centrosome. They are made up of 9 evenly spaced peripheral fibrils of the tubulin protein. They are made up of tubulin protein. Important point, right? And each of the peripheral fibers is a triplet. Now, in the case of cilia and flagella exoneme structure, we have seen that they were the doublets, but here they are the triplets. They are the triplet structures like this. And these triplets are again interconnected to each other. Okay, so each of the peripheral fibers is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and are called as the hub, which is connected with the tubules of the peripheral triplets by the radial spokes made up of protein. So as we have seen in the case of that, they are interconnected with the, in the case of cilia and flagella, they are connected with the radial spokes. So here also the centrioles is also a proteinaceous and they are called as the hub. The proximal region of the centriole is also proteinaceous and they are called as the hub, and that are interconnected with the tubules of the peripheral triplets by the radial spoke-like structure, and they are proteinaceous. Right? The centrioles from the basal body of a cilia or flagella and the spindle fibers that give rise to the spindle apparatus during the cell division in the animal cell. So when the cell divide, right, like this, when the cell divides, these spindle fibers are get attached to the centromere structure of the chromosome, like this. Centromere structure of the chromosome. So what happened? These spindle fibers makes the chromosome movement towards the pole. Each chromatids now move towards its pole in this way. So these spindle fibers are very, very important at the time of cell division. Okay, so the centrioles form the basal body of cilia and flagella, and spindle fiber that give rise to the spindle apparatus during the cell division in animal cell. Okay. Many membrane-bound minute vesicles called microtubules that contain various enzymes are present in the both plants and animal cells. Okay, so glyoxysomes and peroxisomes are some of the organelles that contain different enzymes. Okay, so they are the microbodies. Okay, they are the microbodies that contain various types of enzymes, and they are present in both plants and animal cells. Okay, so if you remember the photorespiration part, that contained the three different organelles, that peroxisome, mitochondria, and chloroplast, right? So there comes the peroxisome. They contain lot many enzymes in it that is responsible for the photorespiration. Okay, so we have studied about the uh, ribosomes in this particular session, and also about the cilia and flagella structure, and centrosome and microbodies. So just for a revision, that about the ribosome is a non-membrane bound structure, right? It is a non-membrane bound structure, and it is present in both prokaryotes and eukaryotes. The prokaryote is having the 70S type of ribosome, whereas in the case of eukaryote, it is 80S type. Now, if you remember, this 80S type is present in the eukaryotic cytoplasm. Okay, eukaryotic cell cytoplasm region. But the two organelles like mitochondria and chloroplast, they also having the ribosomes. But these ribosomes are of 70S type, the one that is present in the prokaryotic cell, right? So this is very important. Inside the chloroplast and mitochondria, DNA are also present. Ribosomes are also attached to the surface of endoplasmic reticulum and on the nucleus. When they are present on the endoplasmic reticulum, they are called as rough endoplasmic reticulum. Okay. So we have completed this session that constitute the ribosome, cilia and flagella structure, okay, the microbodies, and also the centrosome. So this is all about this session. We will meet with one more interesting session. Thank you very much.